So my name is Brinton, Seashore-Ludlow, and I work at Karolinska in the group of the larger group of Olicalionemi. And I guess we'll also hear from some other people in that group later today. And today I'm going to tell you about the high content imaging and patient-derived models. So our goal in our larger group is to use information from patients, often the tissues and the plural tissue and pleural effusion, to understand how patients respond to drugs. And for our patients that we studied several different diseases. We work mostly in ovarian cancer or AML. We get things like clinical data, blood and bone marrow, tissue, and then these pleural and ascites fluids. We then do cellular and molecular profiles and individualized drug screening on these, and we create patient-relevant cells and living biobanks of these. And part of our work goes to developing new tools for the drug testing and analysis. And part of our work goes at integrating all of these types of information on the N=1 and then across patient cohorts. So as we heard a beautiful example from Barend of how this can be used to reposition drugs to patients or to provide actionable drugs for patients who have blood cancers. But we're not in that area right now. We're mostly focusing on how we can use this to either understand what biomarkers could be predictive of response, look at how functional biology in that disease setting, and create data resources for later. So the current testing that we have been doing has been mostly optimized in AML, and there we seed cancer cells into 3D4-well plates. This is an example of a bone marrow that we get. And this, when we do this with cell titer glow, which is a plate-based reader version of the assay, where you measure ATP as a surrogate for viability. We do this across 528 drugs with an emerging clinical candidates. And this is done in 5-point dose response across 8 plates. And we do this with cell titer glow. We get out around 3,072 measurements. And this we use to then understand a patient-specific drug response profile. The way we put that together is by fitting the curve. So we have 5 points for each drug, and we can fit a curve. And we modify the area under the curve to a drug sensitivity response score. And we do this across the whole library, so we get a lot of DSS scores. So we've been doing this in solid tumors as well and building up this capability. And right now I will tell you a little bit about what's going on in ovarian cancer. So ovarian cancer is a group of diseases that stem from the ovary, and it can be diagnosed at different stages. Unfortunately, the most people are diagnosed at the later stage, stages 3 and 4, because the symptoms are quite diffuse. So in general, it comes from 5 or 4 different types of cells, most of the cancers arising are from epithelial cells, but there are other types of cell of origin as well. And then of the epithelial cancers, there's 5 main histotypes, the majority of which are serous. And that can be either classified as high-grade serous or low-grade serous cancer. And in general, all of these patients have, in terms of the epithelial cancers, have different types of common mutations and molecular aberrations, but actually all of the patients get the same treatment. So all of them come in with and get a diagnosis and a histotype, but they're all given, at least in Sweden, the same treatment, which is usually a combination of surgery, so cytoreductive surgery, and then carboplatin. And this is regardless of the fact that they have different driving mutations and potentially different biology that they would benefit from. So what we're working on is understanding how to better treat these patients by understanding what the drug response landscape looks like. So as I said, we get different types of material from the patients. We get blood, and that's what we use to do germline sequencing of the patient. We get tumor tissue, and that's what we use to drug test and biobank and then do genome sequencing on, or sometimes RNA-seq. And then we get something called ascites fluid, which is a buildup of fluid in the peritoneum, which comes from the tumor. So it's basically metastasizing into the stomach, and there's a buildup of fluid there. Stomach area. So using this material, we basically take our tissue that we get from patients or the ascites fluid, and we dissociate it into aggregates that we can plate. And from that, we've been making long-term models, so patient-derived models of the cancer, as well as fibroblasts. And we make sure that our models, this is done by Greta in my group, but make sure that our models are matching the cancer that we have get them from the patients. So here you can see each chromosome up here, and you can see that we have blood. We don't have any mutations in the germline, but we have different patient-derived cells from ascites and tumor tissue 1 and tumor tissue 2 and tumor tissue. And we see that we have good correlation between those. We also look that we have exact changes, so genomic changes, mutations. For example, in high grade, we know that there's about 95% of patients have P53 mutations. So we make sure that the cells that we grow out actually have the same exact P53 mutation to make sure that what we are growing out is relevant. And using this material, we've been able to do drug testing using that platform that I was talking about before with 528 drugs in 5 different concentrations. And this heat map shows the DSS scores, where blue means that it's more sensitive or to the bone marrow cells that we use as a control, and red means that it's more sensitive to the cells that are being shown there. So if we zoom in there, we can see that this clusters the different types of cells that we're looking at. So as I said, we have cancer cells, and the light green show regular cell lines in ovarian cancer cell lines, and the dark green show our patient-derived models. And then we have the light blue, which is over here, is showing healthy bone marrow response, and here is showing the actual fibroblast models that we've developed. There's less variation between the fibroblast models than the patients itself we've seen so far. And if we look at what this actually means, so as I said, this is from the DSS score. These are the actual curves that you get, for example, for tosidostat, cisplatin, which is one of the standard of cares. Depending on where you live, you either get carboplatin or cisplatin, and paclitaxel, which is another standard. So the blue curve, again, the light blue curve is the healthy bone marrow. So that's the response that you don't want to kill the bone marrow cells, but you do want to kill the cancer cells. So we're looking at what is killing them there. So this is really interesting to understand the overall drug response landscape, and you can use it to scale up into drug discovery projects, which we're working on now. But what we can't do is something similar to what Bernd has described, which is do this quickly. So what we've been working on is looking at short-term testing in ovarian cancer. This is where we first designed a drug library that's specific to the ovarian cancer. So we used input from both the gynecological oncologists and surgeons, as well as information from our functional taxonomy that we created before. And we've created a customized drug library that we have around 58 drugs on, including some combinations. And instead of trying to grow models, what we're doing is actually plating the cells directly into ULA plates, which stands for ultra low attachment when they're U-bottom shaped, so that the cells form spheroids or aggregates in the plates. We're then using treating them with drugs and reading out with microscopy. And this is done in planes so that we can understand the size and the growth of those aggregates. This has been done by Emma Åkerlund in our group. After we get out the information, we'll do some live cell imaging first and then fixed cell imaging. So we get an input image that we can then find the area that's actually alive versus the area that's dead, or the cell level, depending on which markers we're using. We then translate this using the same type of approach with a drug response, dose response curve. So look at how the drug responds over the different doses, and can create the same patient-specific drug sensitivity and resistance profiles using this imaging data instead of cell titer glow. And this can help us on the individual patient level suggest compounds that or drugs that might be actionable for the patient. But we're also looking at patterns across the cohort and matching this to genomics and transcriptomic features as well. So this is just an example of the actual images showing a dose response curve, where we can get a dose response curve for both the viable mitochondria in yellow, and then the number of dead cells or cells where you have a permeabilized outer membrane in blue. And finally, we've been doing this across a small cohort so far, where we've been looking both at the ascites fluid and the tumor tissue. This is just showing us what we have, what types of patients we've had, what disease they've had. So we get them when they have potential high-grade serous ovarian cancer, but in some cases it turns out to be later re-diagnosed, or if they're having, or patients that relapse. And this is just the drug response landscape so far. And I can tell you the take-home messages from our study is that we are able to do this with quite good quality control now that we have everything set up. We've done it with good quality control for 13 out of 17 samples. We're doing the same type of imaging approach in 2D as well. And there we have also good quality control. So we think that these approaches are amenable both to the aggregated tumor cells and ascites fluid samples, so we can attest either one. And so far, there's actually quite good correlation between the two, though we do see various changes in certain drugs. And as Barend was saying, the time to turn around, so getting out the answer of what drug is appropriate needs to be quick. And here we can do this in around 3 days as well. So that was on the technical side. And in terms of the biology, so we are able to identify drugs that effectively target the patient's disease ex vivo. And we do see good concordance between targeted drugs and the mutational profile of the patient. So we're looking forward to exploring this more. And these 2 points are both the same for the 2D that we've done with the large drug screen as well. And with that, I'd like to show you just quickly what the diversity of the images look like. So how they grow very differently each patient, which means that for each patient, we need to have a way to characterize how what's still alive. And we need to have a robust pipeline for analysis. So in terms of that, this is our group, and thank you.